Good afternoon, everyone. Today, we will be tackling about a very familiar topic for all of us, the salivary glands.
Salivary glands. The salivary glands are a group of compound excretory glands secreting saliva. Saliva is a complex fluid produced by the salivary gland. The saliva forms a film fluid coating of the teeth and mucosa, thereby creating and regulating a healthy environment in the oral cavity. Structure of terminal secretory units. The basic functional unit of salivary gland is the terminal secretory unit called acini. The terminal secretory unit, irrespective of size and location, is made up of epithelial secretory cells, namely serous and mucous cells. Serous cells. Serous secretory cells are pyramidal with broad base on the basement membrane. The apex faces the lumen. The serous cells have a spherical nucleus placed at the basal region. The apical cytoplasm of these cells shows accumulation of secretory granules. In human beings, the granules contain a dense core or twisted skin-like structure with a lighter matrix. They can be visualized in a semi-thin plastic embedded tissue section, stained with toluidine blue or specific cytochemical techniques. The granules are closely opposed to each other but retain their individuality. Mucous cells. The mucous cell, like the serous cell, is specialized for the synthesis, storage, and secretion of secretory products. However, its structure differs from that of the serous cells. In routine, histologic preparations of the apex of the cell appears empty except for a thin strands of cytoplasm, forming a trabecular network.
We have the myoepithelial cells or the basket cells. They are closely related to the secretory and intercalated duct cells. The body is small, pyramidal in shape, filled mostly with nucleus and has numerous protoplasmic projections, stellate or spider-like with a flattened nucleus, scanty pernicular cytoplasm, and long branching processes that embrace the secretory and duct cell. So we have the ducts. The ductal system of salivary glands consists of hollow tubes connected initially with the acinus and then with the other ducts as the ducts progressively grow larger from the inner to the outer portion of the gland. The ductal system also actively participates in the production and modification of saliva. It can be classified according to location, which could be interlobular and interlobular, and structures, function, and location, which could be excretory, secretory, and intercalated. First, we have the intercalated ducts. It is an interlobular duct and smallest duct that connects the terminal secretory units to the next larger duct, the striated ducts. It is lined by a single layer of low cuboid cells with a relatively empty appearing cytoplasm. Next, we have the striated ducts. It's also an interlobular duct. It receives saliva from the intercalated ducts, from the largest portion of the duct system constituting the interlobular component of the duct system. It is lined by a layer of tall columnar epithelial cells with a large spherical centrally placed nuclei. Lastly, we have the excretory ducts. It is an interlobular duct located in the parting septa. It is located more proximal to oral cavity and has greatest in diameter. It receives the more completely formed secretions. Unbranched excretory ducts are simple and branched excretory ducts are compound.
Next is the connective tissue elements of the salivary gland. It consists of fibroblasts, macrophages, mast cells, occasional leukocytes, fat cells, and plasma cells. The connective tissue of the salivary gland consists of a surrounding capsule that demarcates the glands from the adjacent structures. The extensions of the connective tissue as septa inward from the capsule divide the gland into lobes and lobules. The septa contains blood vessels and nerves that supply the parenchymal components or the glandular components and excretory duct. Plasma cells produce immunoglobulin that are secreted into the saliva by transcytosis. The main immunoglobulin in the saliva is immunoglobulin A, and it also secreted small amounts of immunoglobulin G and immunoglobulin M. And the immunoglobulin A synthesizes as a dimer complex with an additional protein called J chain. In the lobules of the salivary gland, finer partitions of connective tissue extend in the adjacent endpiece and ducts. They carry the arterioles, capillaries, and venule of the microcirculation and branches of the autonomic nerves that innervates the secretory and ductal. For the blood supply, the salivary secretion is predominantly 99% water. Ducts to the level of the intralobular striated ducts supplied with a dense capillary network which loops to the intercalated ducts. The arteriovenous communication are seen in the larger interlobular ducts, and an extensive capillary plexus originates from separate arterioles that exist around the excretory duct. Then the venous follows the arterial supply and anastomosis, which more blood supply is diverted, resulting in increased venous and capillary pressures. It also results to increase in fluid filtration across the capillary endothelium. Nerve supply, the salivary glands are innervated by postganglionic nerve fibers of the sympathetic and parasympathetic division of the autonomic nervous system. Next is the classification and structure of human salivary glands. Salivary glands has been classified in a variety of ways, size and location, which has major and minor gland, and based on location as labial and lingual. Next is the histochemical nature of secretory product, which is namely serous and mucus.
Salivary gland, the largest of the glands, and it also lines the mucosa of the upper aerodigestive tract and the overwhelming entirely of the mouth. Parotid gland, largest major salivary gland, and produce saliva to keep your mouth lubricated and to aid in chewing and digestion. The main excretory duct of parotid gland is Stenson duct, and it emerges from deep within the parotid gland and travels from the anteromedial border of the gland to the anterior border of masseter muscle. Submandibular gland is the second largest, and it produces saliva which moistens the mouth and aids in chewing, swallowing, digestion, and helps to keep the mouth and teeth clean. So the main excretory duct of submandibular gland is the Wharton duct. Originates at the submandibular gland, hilum, and then travels around the posterior portion of the mylohyoid muscle. Sublingual gland is the smallest of the major salivary gland, and it primarily produces a thick mucinous fluid and lubricates the oral cavity, which allows for swallowing, initiating digestion, buffering pH, and dental hygiene. So the main duct of this is Bartholin's glands, secrete fluid that acts as a lubricant during sex. There are 800 to 1,000 minor salivary glands located throughout the oral cavity. Labial and buccal glands, its continual secretion of saliva is important for immune defense by preventing dryness of the oral tissue around the lips and by producing antibacterial secretion. Glossopalatine gland, the glands present in the region of isthmus in the glossopalatine fold are purely mucous glands. Palatine glands are minorly salivary glands located under the mucous membrane on each side of the midline of the palate. Lingual gland, so it can be divided into several groups. The first is anterior lingual glands and the second is posterior lingual serous glands. The anterior lingual glands are present near the apex of the tongue. The ducts open on the ventral surface of the tongue near the lingual frenulum, while the posterior lingual mucous glands are present lateral and posterior to valate papillae and in association with lingual tonsil. Lastly, the von Ebner's glands, one of their roles is to secrete amylase.
So now the development and growth. Salivary glands develop in fetal life as solid buds of oral epithelium. As the buds grow, it proliferates to form coat of cells, and together they later hollow out to form ducts and alveoli. The primordial of the parotid and submandibular glands of humans appear during 6 weeks, the primordial sublingual gland after 7 to 8 weeks, and minor salivary glands during the 3rd month. So the primordial salivary glands is divided into pregland, which takes part in the formation of acini and pre-duct cells, divided into anterior and posterior domain. So this gives rise to the common and individual ducts respectively. So there are 3 stages in the development of salivary gland. The first stage shows the formation of analog and development of the gland. The second stage, further differentiation of the gland, lasts till 7th embryonal month. The third stage begins at the 8th embryonal month and leads to further structural maturation of the gland. Meanwhile, the control and secretion. The physiological control of salivary gland secretion is through the ANS, which controls the volume and the type of saliva secreted. So the secretion of saliva is controlled by sympathetic and parasympathetic nerves, and the release of neurotransmitter from the nerve terminals stimulates the glands to discharge their secretory granules. And this process is called stimulus-secretion coupling. The parasympathetic nerve fibers responsible for creating sear saliva, parotid, and submandibular. So the sympathetic nerve fibers last is the one who produce mostly mucous saliva by sublingual and submandibular.
So moving on to the composition of saliva. Saliva is mainly composed of 99% water and 1% solutes, which includes electrolytes, enzymes, mucus, and antibacterial compounds. The main organic substances found in saliva are secretory proteins, which includes amylase, lysozymes, cystatins, secretory immunoglobulins, peroxidases, lipase, kallikrein, proline-rich protein, mucins, and cysteine. Next, these are the salivary component mentioned a while ago and their specific function. Moving on, the saliva produced by major salivary gland differ from one another in composition. The parotid gland secretes amylase, protein-rich protein, and glycoproteins. Submandibular secretes glycosylated substances, and sublingual gland secretes mucin. Moving on, the total volume of saliva secreted by humans is approximately 750 to 1,000 ml daily, of which submandibular accounts for 60%, the parotid about 30%, and the sublingual about 5% or less, and about 1% of saliva is derived from the minor salivary glands. The most important function of the salivary gland is the production and the secretion of the saliva. Protection of the oral cavity environment is the major function of the saliva. The other functions being assisting in digestion, speech, mastication, taste, and tissue repair.